All right, hi guys. Thanks for tuning in today. Today we're going to be covering visual pathways and the pupillary reflex. So basically looking at how signals get from our eye back into the brain. So we're not going to cover exactly how visual processing works. We're going to focus on the pathway today. So we'll start off just by looking here. You can see this is what we call fundoscopy. So this is looking at the back of someone's retina. And you can see this bit here called the optic disc is where the veins, the arteries, and also the optic nerve pierce into the back of the retina. The key things that I wanted to point to today, though, are this area here called the macula, and the center of that is called the fovea. So the macula is an area where there's no blood vessels, essentially. The fovea is a part of the macula. It's like a little divot inside of it. And basically the important thing there is that we have two different types of light receptors, essentially. We have things called cones and rods. Cones we find inside the fovea. They give us visual acuity and color vision. So they allow us to see really specific things. In the macula more generally, though, we see a lot more rods. They give us essentially our night vision. So they're not very good at perceiving things in a really high level of detail, but they can basically tell if there's light there or not. And you know, you can tell this difference in visual acuity, right? If you're standing in a dark room, things seem a little bit fuzzier than usual. You can't really read things very clearly, but you can still see like a silhouette moving towards you or something like that. You can see your kitchen table if you're sneaking back at night or something like that. But in order to be able to like read the paper that was sitting on that table, for example, you would need light present, and that's because you need the visual acuity that's garnered from the fovea and your cones inside there. Okay, so we're going to look into a little bit more detail now about this, the pathway that we'll focus on rods, but cones use a similar thing. Basically how we turn light signals into a nerve transduction. So we're going to focus on rhodopsin for this because which is in rods because it's just a little bit easier. But as I say, cones follow a similar kind of process. So we'll look first at this dark state. So this is what's happening when you're sitting in a dark room. Now, funnily enough, your nerve cells in a dark room are actually constantly firing, and it's the absence of firing which will result in an action potential essentially going. So first of all, we're going to talk about what's happening in rhodopsin. So this bit here, let me change pens. Cool. So rhodopsin here, this is the protein that's involved in detecting light. It has this thing in it called retinal. And retinal is a derivative of vitamin A. So when your mum or dad always said, eat your carrots because it will give you night vision, there was actually some truth to that. Retinal, derived from vitamin A. Vitamin A is very high in carrots and beta carotene. So basically what happens is in the dark state, retinal is in a certain conformation so that it's not active, and instead that you have these channels here constantly open. Now these are cyclic GMP gated channels, which means that as long as there's cyclic GMP present in the cell, those channels are going to be open, and because it's letting in calcium and sodium, what we're going to see is that this cell is going to be depolarized. And if it's depolarized, it's going to be releasing glutamate. Okay. Now, what is it releasing glutamate onto, you might ask? We'll branch away quite briefly just to give a little overview of this thing called the through pathway. Basically, what that means is that you have your sensor here, which in this case is the rhodopsin, and that sends a message onto this cell here. So rhodopsin, a rod cell, I should say, containing rhodopsin. That then sends messages to a ganglion cell, bipolar cell. Getting ahead of myself. And then onto this one here, which is the ganglion cell. Ganglion cell. The ganglion cell is what will eventually fire the action potential that goes into the brain and tells it it's a signal. So basically the rod is sending signals to the bipolar, and the bipolar is sending cells to the ganglion. And all of these processes are basically saying whether this cell should fire an action potential or not. So that's just branching away. We'll come back to this concept in a second. I'd like to keep going with this for now though. Okay, so that's our dark state. What happens is rhodopsin and retinal are light sensitive. When light hits retinal, it changes its conformation, and that induces conformational change in our rhodopsin protein. That conformational change activates another series of intracellular events. We can summarize this by saying it activates transducin. And what transducin does is it activates this protein here called phosphodiesterase. You might have encountered this before. It's present in nearly every cell in the body, but basically phosphodiesterase converts cyclic GMP back to just normal GMP. What that means is that our cyclic GMP gated channels, which were open here, are no longer open, and so the cell can't keep depolarizing, and so it actually hyperpolarizes. If it's hyperpolarized, that means no glutamate. Okay, so this seems a little bit counterintuitive at first, but basically it means that when there's no light, the rod cell is depolarized, and that means there's glutamate being released. When there's light present, the rod cell is hyperpolarized, and that means there's no glutamate released. Okay, we're going to jump to an extra layer of complexity now. So back to those bipolar cells that we were talking about before, we have two kinds. We have on bipolar and off bipolar. On bipolar depolarize in light. Off bipolar hyperpolarize in light. So in both cases, they're acting to the same stimulus. The stimulus is a decrease in glutamate release. So what that means is that we must have something that's different in the glutamate receptor between on bipolar and off bipolar cells. We'll start with off bipolar because this is a little bit more intuitive. The off bipolar cell has just a simple ionotropic glutamate receptor. What this means is that when glutamate is present, the receptor is open, and so if glutamate's present, then this is going to be depolarized. What this means is, in the presence of light, when we lose glutamate signaling, there's nothing keeping those ionotropic glutamate receptors open, and so they close, and so they'll hyperpolarize. And if they hyperpolarize, then they will stop sending signals to their ganglion cell. With the on bipolar cells, they have a type of receptor called metabotropic glutamate receptor, and they have the mGlu-R6. What this does is, in the presence of glutamate, it inhibits channels. So it's a transmembrane protein, and it's basically a G protein coupled receptor. similar kind of structure. But basically what it means is that with glutamate present, there's secondary messengers and if there's those secondary messengers shut down channels and close channels, which means they're hyperpolarized. Hyperpolarized in the presence of glutamate. Cool. Hopefully you're still with me so far. What this means is that when we get the decrease in glutamate signaling from our rod cell in light, there's less inhibition of these channels. So you get low glutamate then inhibits these processes essentially. And so if you inhibit a closed channel, what you're essentially doing is you're opening the channel. And so you get depolarization of that cell then when light is present. Those depolarized, the depolarized bipolar cells now signal onto the ganglion cell. And similarly to the bipolar cells, our ganglion cells can be on or off. Same kind of thing. Ganglion cells will fire an action potential when light is present. Off ganglion cells will not fire an action potential when light is present. These both have the ionotropic glutamate receptors. So basically, if we're following the course of the on ganglion cell, so the rod cell in the presence of light stops releasing glutamate. So there's no glutamate, which means that our on bipolar cells are able to depolarize. They'll release glutamate through that depolarization process, and that will act on the on ganglion cell to activate that and fire an action potential. With an off bipolar cell, without the glutamate, they hyperpolarize, which means that they stop their release of glutamate. If they're stopping their release of glutamate, then the off ganglion cell just doesn't fire. There's no signal for it to fire because glutamate is the signal for firing. Okay, hopefully that made sense. If it didn't, please post a comment and I'll try and explain it in a bit more depth in another video. But for now, I'm going to take us back to the visual pathway in general. So, a lot of people have seen this before. It can be very intimidating to begin with, but when you break it down, it's not too hard. So starting off, we've got the right eye and the left eye. We're looking at it in the radiographic plane, so as if we're looking up through the feet of the patient. Pupils here. I'm sorry, I'm no artist, but hopefully you get the drift. Basically, each half of the visual field is perceived by a different kind of set of tracks in the optic kind of pathway. So our nasal fibers, shown here in blue and grey, actually pick up the temporal field of view, because the temporal field of view starts out here, comes in through the eye, and will activate the nasal fibers. And likewise, the nasal field of view will come in and activate the temporal fibers, in this case green and red. Okay, so going over that again, blue is our right nasal fiber, and that is detecting our right temporal field of view. I'll just give you an example of each. We won't go through all of them, but likewise, our left temporal fiber has got our left nasal field of view. Okay. What happens then is, so this bit here, this is the optic nerve, so the optic tract, pretty standard. So it exits the back of the eye, and then when it's moving through the brain, we get this partial decussation. So we get the nasal fibers of each eye cross over in this thing called the optic chiasm, whereas the temporal fibers continue running ipsilaterally. I hope that makes sense. So ipsilaterally. Some key anatomical landmarks that I'll point out just at this point now. We have the internal carotid artery sitting on either side of the chiasm. So if for some reason we were to get a rupture or some kind of swelling, vasodilation in these vessels, you'd get compression of the optic chiasm. Sitting inferior to the optic chiasm as well, right in the middle here, is our pituitary gland. So if we have an adenocarcinoma of the pituitary gland, there is the potential for it to put pressure on the optic chiasm as well. We'll come back to those a bit later. The first stop that all these, the optic tract kind of goes to is the lateral geniculate nucleus in the thalamus. So if you're remembering basic principles of neuroscience, the thalamus is the relay center. So things come there to get processed before they get sent to other parts of the brain. Now, because of this partial decussation in the fibers, our left lateral geniculate nucleus is picking up the signal from our right temporal field of view and our left nasal field of view. These bits together, you can imagine if we have our field of views like this, so right eye, left eye, our right temporal field of view, and our left nasal field of view. So you can see that these would come together to give us our right field of view. Essentially, it's everything that you can see on the right half of both of your eyes. That's processed in the left lateral geniculate nucleus. Likewise, on the right side, for the right lateral geniculate nucleus, that processes the left field of view because of those crossing over, that crossing over. From the geniculate nucleus then, we then get the optic tracts, which kind of fan out from the back of the nucleus to our primary visual cortex, which we call V1. This is, in this area here, that's where you actually get the processing of visual information, integration with sounds and with taste and with smells and all those kinds of things. And then that will send more information up to your secondary and tertiary visual processing centers, visual cortices, and yeah, from there you'll be able to actually interpret what you're seeing and be able to figure out that the thing that you're looking at isn't just a red sphere, but it's actually an apple that could be really tasty and nice to eat. Cool. Now, I'm not a very good artist, so to try and just give you a bit more of an appreciation of what these optic radiations look like, you can see that here, so this is our lateral geniculate nucleus again. They fan out over quite a large area of the brain. This has quite significant clinical consequences because it means that if we have a small infarct, we can get very specific patterns of visual loss. And I'm cautious of time, so I might explain our patterns of visual loss another time. I do have them here. I think I'd like to make a separate video of that though, to go over that in a bit more detail. So keep posted for that. I will definitely come back to that. But just to get this appreciation now of the optic radiations and how they cover a large area, so they're going to be supplied by a few different vessels as well. So as I say, I will come back to this slide, don't stress. What I'd like to cover now quickly though is the pupillary reflex. So what this reflex is if you shine a light in someone's eye, you'll notice that their pupil will constrict, but it's not just the pupil that the light shines in that will constrict, it will also constrict the other eye. So that might not make a lot of sense. I'll explain that again. Say you've got a torch here. As I say, I'm not a great artist, but you get the idea. Torch shining into your right eye. What you'll notice is that your right pupil will constrict. What we call that is the direct pupillary response. What will also happen though is if you shine your torch into the right eye, but look at the left eye while you're doing so, the left pupil will also constrict. We call that a consensual pupillary response. These are thanks to these special types of ganglion cells, which I've done here in pink. Basically what they do is they, I have the name here for you. They are the intrinsic photosensitive ganglion cells. They're special ganglion cells. They don't rely on photoreceptors to detect light. And they're involved in non-vision related information. So whereas these fibers here in our retina were sending information back to our visual cortices, our IPGCs, intrinsic photosensitive ganglion cells, they are not involved in that. They're involved in things like your circadian rhythm, detecting light on a more general scale, all that kind of thing. Basically what happens is they send fibers still with that optic track. They remain ipsilateral at the optic chiasm, and then they actually leave the optic tract before it reaches the lateral geniculate nucleus. So they branch off into this area here. We call that the pretectal nucleus, this area here. You have one on both sides. So this one here is our right pretectal nucleus. The pretectal nucleus then sends out a branch to both, what we call them the Edinger-Westphal nuclei. So you have, again, one on each side, the EW nuclei. So it doesn't matter, this is paralleled on the opposite side. Fibers from your pretectal nucleus will innervate both EW nuclei. The EW nuclei will then send a parasympathetic branch to a ciliary ganglion on each side. The ciliary ganglion will then supply another parasympathetic nerve to the sphincter pupillae muscles in the pupil. So these guys here, that's our preganglionic parasympathetic fiber. This is our postganglionic parasympathetic fiber. This here is the ciliary ganglion. We're innervating sphincter pupillae. So this guy here, that's our right pretectal nucleus. It's projecting to both our right and our left Edinger-Westphal nuclei. And from there, sending fibers out to the ciliary ganglion. So you can see here now, if you had light coming in from one eye, it would travel down and innervate a response that's going to affect both eyes. That's the basic idea behind the pupillary response. Thank you guys for staying with me. I know this was a long video. Keep an eye out on the channel for me to explain the visual defects pathway. And if you've got any comments or feedback, I'd love to hear it. So give me a shout.